Today we're going to continue talking about hearing science. We're going to go into the inner ear where the frequency analysis happens. So just to orient us in the context of our last lecture where we talked about an overview of the landmarks and the functions all throughout the auditory system, today we're really going to focus on the basilar membrane. We're going to understand the topic of tonotopicity and the nonlinearity of frequency processing. And we're going to spend some time talking about the functions of the outer hair cells. So the general function of the cochlea is to analyze different frequency components of a complex signal. We touched on this a little bit in the last lecture, but this is just a reminder that for all those frequency components, what we need to do is figure out the mechanical motion that has been set into place by the acoustic wave. And then the hair cells convert that to chemical reactions, and then those chemical reactions give rise to an electrical pulse that's ultimately sent toward the brain. So again, just as a reminder, if we have a complex tone consisting of a number of different frequencies like this, what we essentially want to do is break that apart into its constituent components. So musicians will recognize this as, for example, identifying the notes of a chord. So even when you don't have something as sophisticated and nice as a chord, any complex sound has multiple components that we want to keep track of separately. So essentially what we're describing is a spectrum. So I want to leverage this analogy that we introduced in the last video about coin counters. So as opposed to the one that you'd find in the bank or the grocery store where everything happens sort of behind the scenes, there are other ones that you can purchase that you actually can see everything in action. So the goal here is to think of this as sorting by a particular feature of the input. Okay, and so you can imagine sorting coins by color or sorting them by year. And so in this case, obviously it makes sense to sort them by size because the size indicates the denomination. So, we have a tiny little hole for the dimes and a slightly larger hole for the pennies and so forth up to the larger quarters. So imagine that instead of size of the hole for a coin, we think of this as size for a waveform, where we can increasingly make this a longer and longer period for our waveform. If we divided sounds in this way, we'd essentially be dividing the sounds by frequency. And so this is really the principle of tonotopicity. So the idea is that all throughout the auditory system, starting in the basilar membrane, the different frequencies will be represented at different places. And although this might seem totally normal and intuitive for some of you, I want to remind you that, it could have been divided in different ways, right? So suppose we had a different set of neurons for loud sounds and a different set of neurons for medium sounds and a different set of neurons for soft sounds. That would have been, say an intensity topic scale, but instead we're organizing things primarily on the basis of frequency. So as we look, as we look at the different parts of the basilar membrane, each part is going to have a characteristic frequency that belongs there. So we want to think of this as unrolling that cochlea. Okay, so it's just easier to think about this on a straight horizontal diagram rather than looking at it as a spiral. And so you can see that, that different places of excitation along that unrolled cochlea correspond to different frequencies, which we can see here in the fine structure as, a fast repeating wave, a medium repeating wave, and then a sort of slower low frequency wave there. So this is a diagram that I want us to spend a decent amount of time on. So what I want you to notice is that we have excitation patterns here. That's what we're calling this. The colored line in the middle of this diagram is the basilar membrane. And what we're viewing in the color scheme is how much excitation there is. So the closer to red it is, the more energy you have. And the closer to blue it is, the less energy you have. So we can see that as this is rolling along, there's a particular place that has this displacement of the basilar membrane. So you can think of this as like, say, dropping a stone into a pond, and you can see those ripples forming out. These are those ripples that we're seeing. It's a traveling wave that's propagating throughout the cochlea. Now, there are three things I want you to keep in mind about this diagram, and these are the three, I would say, the most important things to think about as we describe the excitation pattern. The first is tonotopicity. So those different frequencies have different places. The 10,000 hertz tone is the closest to the stapes. This part of the diagram here represents the tympanic membrane, the ossicles, and then right away you have the place for the highest frequencies, and then farther away you have the medium, and then finally the low frequencies here. And so just to zoom in on this here, the second part is the asymmetry. So you'll notice that the color is different to the left of the peak compared to the right of the peak. So leading up to it, we have activation. We finally get that peak of excitation right here. And then right after that, there's nothing. And so this pattern, you'll notice, is asymmetrical. It doesn't approach red and then gradually fade from red to blue. It reaches its peak and then immediately goes down to basically no energy. So that asymmetry turns out to be quite important, and will explain a lot of different interesting details about how our hearing works. Finally, we want to look at the fine structure, the shape of the wave internal to that traveling wave that's propagating along. So for the lowest frequency one, we can see that this fine structure really represents a low frequency wave compared to a higher frequency one, which oscillates at a more rapid rate. And then finally, the really high frequency one, we can see has just sort of a shorter period or shorter wavelength there. So although these aren't drawn exactly to scale, what it's trying to represent is that not only do the frequencies have their own individual place along the basilar membrane, but at that place, they're also reflecting the frequency and how fast the oscillation occurs. Now, the general theme here is that the highest frequencies are at the base of the cochlea and the low frequencies are at the apex. And so this tends to trip some people up as they're first learning, because you think of bass and you think of like bass notes on a guitar. So we're not thinking about that. We're really thinking about bass in terms of structure. Like a tree has a base and an apex to it, and so too does your cochlea. Your cochlea has a base and an apex, and at the base, that's really where the excitation is starting, and up toward the apex, the top of it, that's the end of the line, and that's really where the low frequencies will be found. There are a lot of different diagrams of these principles, and so you'll find, in any, Google image search about the basilar membrane or the cochlea, you'll find images like this. So I encourage you to find one that really clicks with you, and hopefully they'll all represent some of the things that are really central to these principles. Now, what you'll see here is representation of tonotypicity and the asymmetry, because you'll notice, for example, here in the 100 Hertz diagram, it sort of goes up and then immediately drops down after the peak. But what's not represented here is the fine structure. So make sure that we commit to memory those three principles of tonotypicity, asymmetry, and fine structure, even as we're viewing and critiquing different diagrams. So here's another diagram that actually reflects the true nature of the shape, which is that this really is a spiral as it goes throughout the cochlea. We have the tonotypicity represented here in that the highest frequency is toward the base, the middle frequency is the middle, and then the lowest frequency is at the apex. I wouldn't say that the asymmetry is represented here, though, so that's something that is maybe not as clear-cut in this diagram. There are some, images out there that are actually animated. So what we want to highlight here is the fact that this traveling wave works up to its area of peak excitation, and then it all dissipates, right? It immediately settles down after that peak. So this is different than how it would work as if you, for example, dropped a stone in a pond, in that you'd see ripples going all the way to the edge of the pond. Here, they find their spot and then they stop. So, and this is represented in our animated diagram down here as well. So this shows the asymmetry. We have a little bit of excitation before the peak. The arrow here is pointing at the peak, and then we can see the fine structure as it actually reflects the ups and downs, which are just a direct consequence of the tympanic membrane moving in and out. The way that we want to understand frequency perception should be reminiscent of how we understood sound intensity, in that it's logarithmic. So just a quick reminder, logarithmic means that we're referring to a proportional difference rather than a linear or absolute difference. So for example, we would express things not for example, like 100 Hertz more than some frequency, but say twice as much, or a third as much, or 13% more, or 70% more. So let's walk along what this might look like here. So what we have here is a diagram of the frequencies, and I've drawn them on a linear scale. So for every tick mark we see on the axis, that space is worth 1,000 Hertz. And on the y-axis, we see in the cochlea, where that would fall relative to the apex. So for example, if we take this range that goes from 0 to 2000, that could correspond to that part, that range of the cochlea. And the next range of 2000 hertz could correspond to that part. And so this is what it would look like if we had a linear scale, where the equivalent amount of 2000 hertz would correspond to an equivalent amount of, say, 5 millimeters on the cochlea. And so we'd have a linear line just connecting those. So this is not how it works. This is just an illustration of what linear scaling would look like, where 2000 hertz just corresponds to an absolute amount of size in the cochlea. Instead, what we have is something that looks like this. So this is a logarithmic curve. And what it shows us is that there's a lot more cochlear space devoted to representing these lower frequencies and a lot less space devoted to the upper frequencies. So let's think about this logarithmically. So let's look at between a 1000 and 2000. It's a difference in 1000 hertz, sure, but we can think of it as a doubling. And so in terms of cochlear space, we see about 4-ish, 5-ish millimeters representing that there. And we can follow this through by following this tick up and seeing where it intersects the y-axis. And then at 2000, it intersects just before the 20 mark. And then for the next doubling, right, we're going from 2000 to 4000. We again have about 5 millimeters of space. And then for the next doubling from 4000 to 8000, we have another 5 millimeters of space. So what we're seeing is not that millimeters refer to a specific number of hertz, but they refer to a specific proportion of sound. So this is just those that same diagram rather, but now all we've done is change how the x-axis is laid out. Now instead of each tick mark referring to a specific number of hertz, in this case 1000, each tick mark on the axis now refers to a doubling of the last frequency. So we're starting at 125, we double it to 250, we double it to 5000, we double it to 1000, and notice how this diagram now looks more like a straight line, because now equivalent amounts of cochlear space correspond to equivalent proportional differences in frequency. Another way of viewing this is like on a spectrum, right? So here we have linearly spaced frequencies. They start at 200, and then the next one is 400, 600, 800, 1000, and so on. So they're all spaced exactly 200 hertz apart. Now, if we converted the x-axis scale just to show us these frequencies logarithmically, we'd push them over like this. And so the numbers on the axis scale now are quite scrunched together, so we can just space them out a little bit differently. And notice how, as we represent things logarithmically, even though they're all 200 hertz apart, they look to get closer and closer together the higher you go. And that's because 200 hertz relative to 200 is a big difference. That's a 100% difference. But 200 hertz relative to 4000, that's a tiny difference, right? So that's represented accordingly as a tiny space on the graph. So here we have just those same frequencies, right, on this scale on the left, right? But in the cochlea, if we were to scale frequencies that would be equally spaced, it would look more like this, where we'd have 125, 250, 500, and so on, and that would actually space them roughly equally apart in the cochlea. If we took those frequencies that were all linearly spaced apart by 200 hertz each, this is how they would sound. And on a spectrum, it would look like the graph we have on the top. Now, in the cochlea, conversely, it would be laid out like the graph we have on the bottom, where the lowest frequencies would be farther spaced apart and the higher frequency components would be smushed together. Here we have a different arrangement of frequencies, which really respects the arrangement in the cochlea. So this doesn't reflect any natural sound that you would hear. This is really something I just generated through a computer. But suppose we had sound frequency components that were, in fact, successive doublings of each last one. So this one, begins at 125, 250, 500, 1000, and so on, and it would sound like this. So it's certainly different than the tone complex we heard before. It doesn't have one specific frequency to it. We can actually hear out the different components, especially if you're a musician. And the spectrum of this would look like what we have up top here. So this doesn't look like a spectrum of any speech sound we've heard before, because the lower frequency components are very close together, and then the higher each one goes up, the farther it is from the previous one. So this just reflects what frequencies would space out like if they were in fact logarithmically spaced. So what we see here is just what that transformation looks like. So the graph on the top and the bottom both cover the same range of frequencies. It's just about how much we're warping what that axis looks like. Now the axis is still monotonic. It's still a fair representation. But you can see that the lower frequency region is expanded out and represented and what we call foveated. It's zoomed in so that we can really see all the differences, whereas the differences in the higher frequency region are really smushed together because they're not really as differentiable by our ears. So speaking of frequency differentiation, here's an example of how we benefit by thinking proportionally rather than linearly. So suppose we have a sound of 500 hertz and then another one at 505. So the difference between those you can say is 5 hertz, but you could also express it as 1%. And so these two frequencies, 500 and 505, are as different from each other as 1000 and 1010. So it would not be right to say that 500 and 505 are just the same as 1000 and 1005, because 1000 and 1005 is a 0.5% difference. So the question, can you detect a 5 hertz change in a frequency, really is not the way you want to phrase the question, because it always depends on the reference frequency. It also depends on other things like the spectral composition of the signal, how complex it is. But we really want to start thinking about percentages or proportions rather than absolute numbers. So for sounds that have harmonic content, like the ones that we typically work with enough practice, we can discriminate really fine differences on the order of 0.2%. And if we don't have that harmonic information, and this is something we'll talk about later as we get into pitch perception, then we need more of a 6% change. So that's a pretty stark difference there. But with all the information that's normally available, we're very, very good, in fact, better than other kinds of animals at distinguishing differences in frequency. An important part of understanding what's going on in the inner ear is understanding hair cells. And just to make sure we're clear on this, these are not the hairs that are in your ear canal. These are really, really tiny cells that are in the internal structures in your ear that you wouldn't be able to touch with your fingers. So these are cylindrical cells that have cilia on top, and those cilia are sensitive to the motions of that vibrating wave, that traveling wave that we illustrated earlier. The outer hair cells receive that motion and actively shape and amplify it in a way that we'll illustrate here in just a few minutes. And then the inner hair cells, we won't talk about them so much in this lecture, but the short story is that they'll actually initiate those electrical signals that are eventually sent to the brain. Now, to get a sense of the inner hair cells, first we got to start out and then zoom in. So here we have the cochlea, the spiral-shaped organ. Now we've cut away a section of it so we can see the inside. And suppose we just flatten out that image. What we're going to do is zoom into one of those chambers, and we can see that it in fact can be divided into 3 subchambers. So that internal one there, what we call scala media, the middle one, can be understood and unpacked into this set of interesting cells and organs. And these right here in the middle, those 3 right there are our inner hair cells. So we have zoomed in to a tremendous amount of detail. And then each one of those cells has cilia on top that is sensitive to the motion. So we're not going to spend too much time talking about all the anatomy of this. There are other classes devoted to that. What we want to get a sense of are the 2 major functions of outer hair cells, which could be simply expressed with these 2 sentences. Outer hair cells amplify low-intensity sounds, and they sharpen frequency tuning. And so in order to have these benefits in your hearing, you have to have healthy and active outer hair cells. So outer hair cell loss is a pretty common form of hearing loss that can result from noise exposure or different kinds of medications. And so if you do damage those cells, you're going to lose these 2 functions. So what does amplification of low-intensity sounds refer to? Another word for this that's a little bit more simple is called compression. So suppose we have this input-output function. These numbers are just totally arbitrary. What we want to think of is comparing a linear system to a compressive system. A linear system means that for an equivalent amount of change in input, you get an equivalent amount of change in the output. This is a pretty simple system. So instead, we're going to have a compressive system like this. Now, why would we do this? The idea is that we're bumping up those lower levels, and those would be the soft sounds. We're amplifying those so that those are higher, and then the ones on top aren't really changed as much. And the reason we're doing this is that in the internal part of the system, the amount of change that the neurons need to detect, or in this case, the outer hair cells need to detect, has some threshold. It has to be at least some level. So suppose it needs to reach an output level of 50 before it's even detected. That means that we have to bump up those low intensity sounds in order to reach that threshold. So we have a pretty large input range, and without this extra amplification of the low intensity end, then that whole section of the input range would not be audible. So we have to think about how to lift that up, because there's a lot of interesting and useful low intensity sounds out there. The way we want to think about this is mapping an input range to an output range. Now we have this whole large range of outputs, but what we want to do, because we have that threshold, is squish that range so it's only compressed into the output range that would actually work and fire the cells. So we want to think about this as compression. And now I know that this can seem a little counterintuitive, because compression usually means you're pushing down on something. But imagine that this hand and this spring were just upside down. You can also compress it upwards. And that's the kind of compression that we're talking about in the inner ear here. Because what we're doing is accommodating the fact that those inputs that are lower in intensity would essentially wipe away half of our input range. So we're taking that lower half and compressing it upwards so that we don't only deal with this higher level of sound. I mean, we can still hear them, but we don't want to just restrict our hearing to those high levels of sound. We in fact want to hear those lower levels as well. So what we want to do is map that large input range, compress it up so that that's the whole range that's being expressed. So there are a few ways of thinking about dynamic range that I want to offer here. So for example, you can ask the question, what is the range of possible output representations? And in this case, because we're thinking about really tiny and delicate structures in the inner ear, this range is going to be pretty small. There's not a whole lot of range of movement in there. So we want to compress the representation of the sound. We don't want to compress the sound itself. We just want to compress the representation of it. So another way of asking this question is, how much sound input can be represented? And we want to maximize this. We know we can hear loud sounds, but if we can maximize the range of sounds we can hear, that means we can extend this downward and also hear the soft sounds as well. So our goal is to represent a large range of inputs using only a small range of outputs. And that's why we call this compression. The other main function that we mentioned before was not compression, but frequency tuning. So let's illustrate frequency tuning here. Now, if we laid out the different frequencies horizontally the way we do in a spectrum, and we had broad tuning in the cochlea, this would mean that for those two frequency peaks that are being represented there, they correspond to a pretty wide and indeterminate range of frequencies. So it's not really clear which one or which two frequencies are being stimulated. But conversely, if we had sharp tuning, this would mean that we're pretty sure about exactly which frequencies are being stimulated. And so these frequencies can be harmonic frequencies or formant frequencies. The idea here is that we want a sharp representation. And so instead of having that kind of blurry representation up top in orange, we can have a sharper representation down there in blue. Now, if we view different speech spectra, we can view what the consequences are of having different kinds of resolution. So we have the best resolution on top in blue, the poorest resolution on the bottom in red. And just to zoom into this, in the better spectra resolution illustration here, we can see all those details. We can see the general shape. We can also see the frequencies of all the individual harmonics. And this could be very useful for hearing pitch and other details in the sound. Whereas if we had poor spectral resolution, we could get the general shape of the spectrum and know that there's a peak somewhere above 2000 Hertz, but it's not really clear exactly where that peak is. So when we use the word resolution, we're really using it in the same sense as you'd use for like a TV or a computer screen. And just like resolution on a TV, it's not really about brightness. And for sound, it's not about loudness. We're talking about clarity. How differentiable are the different components? So sharp frequency tuning is important because speech sounds are contrasted by different frequencies. So without sharpened tuning, we wouldn't be able to tell those sounds apart. Recall that in our speech acoustics segment, we knew that differences in place of articulation really boil down to different places corresponding to different frequencies. So if we couldn't hear those frequencies, we'd make a lot of mistakes in determining place of articulation when we're listening to words. So without those healthy outer hair cells, you might know that speech is there, but you wouldn't understand it very well. So looking ahead to, things like quizzes and exams, imagining that you're being asked, why are outer hair cells important for the perception of speech? In the past, I've gotten some answers that weren't really that satisfying. So for example, because they sharpen frequency tuning. What this tells me is what the outer hair cells do, but this answer doesn't tell me why it's important for the perception of speech. Similarly, if you write, because speech requires sharpened frequency tuning, that's true, but what does that have to do with outer hair cells? What I want you to do is pull these two pieces of information together. For example, because speech sounds differ by their frequency content, and outer hair cells give clear access to those frequencies. That's why it's important. You're really connecting both of those things. You could be a little bit more lengthy with this. Sharpened frequency tuning is important because many speech sounds are contrasted by frequency. Without sharp tuning provided by the outer hair cells, we wouldn't be able to tell those sounds apart. So what this is demonstrating is that you know that different speech sounds require frequency tuning and that the outer hair cells are the things doing that frequency tuning. So just pulling this back and recognizing both of those functions on the screen here, we're talking about frequency tuning, which of course is important for speech, and we're also thinking about hearing soft sounds. So without those outer hair cells, we wouldn't get that compression, that amplification of low intensity sounds. And so we would just not know that they're there. And so it's not the reduction of volume for all sounds. It's not just like turning down the volume knob. What it is keeping the loud sounds loud, but just dropping the soft sounds altogether. And so this is one of those things that's pretty misunderstood about having hearing loss, is that it's not that all sounds are softer, it's that the soft sounds are not there and the loud sounds are still there. Another thing I wanted to touch on here, just because it tends to be a pretty common mistake on exams, is that without outer hair cells, it's not that we're selectively losing low frequency sounds, it's that we're selectively losing low intensity sounds. So hopefully this will give you a pretty good overview of the main things we want to know about the outer hair cells and the function of the basilar membrane. We can go on and on about this in further detail, and in fact there are whole classes and whole lines of work devoted just to the function of the auditory system. But just to recap here, we're thinking about tonotopicity, asymmetry, and fine structure on the basilar membrane, and then underneath that basilar membrane we have outer hair cells which amplify low intensity sounds and sharpen the representation of frequency.